my sort of interest evolved during the course of my career, and I became more and more interested in behaviour, particularly how early development might alter behaviour. And it was with a very productive collaboration with Trevor Robbins at Cambridge that we began to work on the isolation reared rat, and that is a very simple model that involves either rearing rats in groups of 5 from when they're weaned, and they are socially reared rats, and rats are social animals, or you have them individually housed, and those are the isolation reared rats. There's nothing else, nothing is different. They're all in the same room together. What the isolates cannot do is touch other rats. And that simple procedure within a period of 4 to 6 weeks produces long-lasting, well, actually irrevocable changes to the structure and organisation of the brain, to their behaviour, their transmitter function, and it gives a sort of insight into how social factors and environmental factors can have profound influence on the development of the brain that in the long term may lead to adult mental health issues. The majority of my work has been on experimental animals, and in particular the rat. In later years the mouse became more important because of the development of transgenic models of various receptor systems that I was interested in. And these models have been very important in improving our understanding of how the brain works. I think there is a real problem when you talk about animal research and trying to model a specific disease. So I think it is rather difficult to talk about a rat model of depression. And that is why I have really concentrated on models such as the isolation reared rat, where what we're trying to do is not specifically model a disease, but to try and understand the events that may occur within the brain when the brain is subjected to environmental challenges. And you only have to think of the terrible damage done to the Romanian orphans in the studies from the Institute of Psychiatry in London to see that there is a parallel between what we see in the isolation reared rat and what was seen in the brains of those orphans. So I think that you can validly say that there is real information obtained from the experimental animal, but I think you have to be careful in your interpretation, and in certainly do not try and over-emphasize the translational value of the information in terms of specific diseases.